Have you ever really stopped to think about how we measure things we literally can't see? Well, today we're diving headfirst into that microscopic world. We're going to answer a really critical question. How in the world do scientists count invisible microbes? And why is getting that number exactly right so important for our health and safety?
Okay, so let's just jump right in with a question that, hits pretty close to home, especially if you've ever had that moment of doubt about the food you're about to eat. It sounds like it might be a complicated answer, but it really boils down to a specific and kind of shocking little number.
40. That's it, just 40. We're talking about the approximate number of salmonella cells that can be enough to give you a really, really bad case of food poisoning. I mean, think about that. It's a ridiculously small amount, totally invisible.
And this number isn't just a fun fact. It's a hard and fast rule that health officials and the food industry live by. Being able to accurately count something that small is, well, it's a huge scientific challenge, and it's what keeps our food supply safe.
But this whole invisible numbers game, it's not just about the food on your plate. Oh no, it's happening all around us, in hospitals, out in the environment, and yep, even right on our own bodies. The line between being perfectly healthy and being seriously sick can actually be a matter of microbial math.
And this slide? It illustrates that point perfectly. Look at the left side. That's normal. 1 or 2 cells of Staphylococcus aureus on a pretty big patch of skin. Your body doesn't even notice. But then look at the right. That number explodes to 10,000 cells in a tiny, tiny square millimeter. And that is the difference between having normal bacteria on your skin and having a full-blown serious infection.
So, before you can even think about counting a specific microbe, you've got this massive problem to solve. Your sample, doesn't matter if it's from food or skin, is like a giant chaotic party with millions of different microorganisms all mixed together. So how do you find and count just one type in that crazy mix? Well, the answer is a concept that is absolutely fundamental to microbiology. It's called a pure culture. The whole goal is to grab a single individual bacterial cell and let it grow and multiply all by itself. What you get is a colony where every single cell is a perfect clone of that first one. It's kind of like finding one specific person in a packed stadium and giving them their very own section to fill up.
Now, what's so cool about this is just how simple and elegant the process is. It's called the streaking method. You start with a big smear of bacteria, then you take a sterile tool, you drag it through that smear and onto a clean part of the plate. You're physically thinning out the crowd. You do this a couple more times, and by the end, you've spread those cells so far apart that individual bacteria are all alone, with plenty of room to grow into their own visible colonies. It's pure genius.
All right, let's move on to the next big question a scientist has to tackle. You've got your sample, maybe you've even isolated what you're looking for. But now, you have to decide, what are you actually counting?
And believe me, this isn't some philosophical question. It's a really practical one with major consequences. Think about it. A dead salmonella cell can't make you sick, but a living, growing one definitely can. So this distinction, it's absolutely vital, and it leads to 2 totally different ways of counting.
This slide breaks down that choice perfectly. A total count is like taking a census. It tells you every single cell that's there, dead or alive. But a viable count, that's the one that really matters for safety. It tells you who's alive, who's active, and who could potentially cause a problem. One is just a head count, the other is a threat assessment.
So, with these 2 different goals in mind, total versus viable, how do scientists actually get the numbers? I mean, they're not just squinting into a microscope and pointing. They've got a whole toolkit of really clever methods designed specifically for the job.
This here is a classic method, and it's all about one simple idea: dilution. You take your sample, you dilute it. Then you take that diluted sample and dilute it again and again and again. You spread these different dilutions onto agar plates. On the plates that are really diluted, you'll finally get a small enough number of living cells that they form separate, countable colonies. You just count those, do the math backwards, and bam, you can figure out how many colony-forming units were in your original concentrated sample.
And of course, today the toolkit is way more high-tech. We have things like qPCR, which can count the copies of a specific gene to give you a total number. To see who's alive, scientists can measure things that only living cells make, like messenger RNA or ATP, the cell's energy molecule. And then you have something like FACS, which is just amazing. It uses lasers to literally count and sort individual cells, one by one.
So let's bring this all back home. We've seen how scientists can isolate, define, and count these invisible little organisms.
But why does all this incredibly detailed work matter so much in our day-to-day lives?
Well, when you get right down to it, the science is the invisible foundation for huge parts of our modern world. It's what ensures the food we eat is safe. It's how doctors can tell you if you have an infection. It helps scientists check the health of our oceans and soil, and it lets industries know if their products are clean and safe. This invisible numbers game really is the bedrock of public health.